Staphylococcus. According to Burgess Manual, Kingdom Bacteriae, Phylum Firmicutes, Class Bacilli, Order Bacillales, Family Staphylococcoxaceae, Genus Staphylococcus. Staphylococcal group of organisms are gram-positive cocces in clusters or in groups. They are catalase positive. They can produce catalase enzyme, so they can break hydrogen peroxide to water and oxygen. They are facultative anaerobes. They like oxygen. Without oxygen also they can survive by fermentation. They are oxidase negative. They can't produce cytochrome C oxidase enzyme. The medically important organisms in staphylococcal group are Staphylococcus aureus, Staphylococcus epidermidis, Staphylococcus saprophyticus. Catalase test. We can watch a small video of catalase test. So we have to take a glass slide. On the glass slide, put few drops of hydrogen peroxide, and to this hydrogen peroxide take a loop, inoculating loop, sterile it with Bunsen burner, burning on Bunsen burner, and take few colonies of Staphylococcus organisms from the culture plate, few organisms, and inoculate that few organisms onto hydrogen peroxide. So when you inoculate, if the organism is catalase positive, you can see air bubbles coming out. The hydrogen peroxide is broken down into water and oxygen with the help of catalase enzymes secreted by those organisms. In catalase negative organisms, if you inoculate catalase negative colonies, there won't be any oxygen or air bubbles are not seen. So that means the organisms are not producing catalase enzyme. This is a gram-stained smear of staphylococcal organisms. You can see the staphylococcal organisms are round in nature and they take blue color or purple color, and you can see in bunches or clusters. Bunches. Staphylococcus classification. Staphylococcus organisms are catalase positive. They can produce catalase enzyme, and on gram-stained smears they look clusters in bunches. These are classified into two groups. One is coagulase positive group. In coagulase positive we have a Staphylococcus aureus organism, and this Staphylococcus aureus organism is beta hemolytic, and on blood agar they produce yellow color or golden yellow color colonies. And Staphylococcus group of organisms always ferment mannitol. They are mannitol positive when compared to coagulase negative Staphylococcus. So non-coagulase organisms in Staphylococcus, one is Staphylococcus epidermidis, and this Staphylococcus epidermidis is novobicin sensitive. On novobicin containing culture media, these organisms won't grow. These are beta hemolytic. Majority of the Staphylococcus epidermidis organisms are beta hemolytic, but few organisms are gamma hemolytic. And another non-coagulase Staphylococcus organisms is Staphylococcus saprophyticus. It is novobicin resistant. On novobicin containing media, these organisms grow, and they are always gamma hemolytic. There is no hemolysis on blood agar culture mediums. Our lecture mainly concentrates on Staphylococcus aureus in this video. Staphylococcus aureus are coagulase positive. They can have a coagulase enzyme on their surface which can mix with or which can associate with prothrombin, which can make staphylothromboin. So this coagulase enzyme can protect this organism from phagocytosis of the host or human immune system. They produce golden yellow color colonies on blood agar culture medium. They are beta hemolytic organisms, and they produce golden yellow color pigment adjacent to the colonies. They are non-motile, non-spore forming organisms. They are facultative anaerobes. They ferment glucose and ultimately produces lactic acid. They ferment mannitol, and this mannitol is the main identifying feature, differentiating feature between Staphylococcus epidermidis and Staphylococcus aureus. So this is the picture of blood agar culture media where the staphylococcal organisms are grown here, and adjacent to this organisms colonies you can see an yellow color or golden yellow color pigmentation for Staphylococcus aureus, whereas epidermidis or other saprophyticus organism, they grow as white color colonies without having any golden yellow color pigmentation around them. This Staphylococcus epidermidis, some organisms are beta hemolytic, some are gamma hemolytic. This is Staphylococcus aureus colonies on blood agar culture plate. You can see golden yellow color pigmentation around the colonies. And on the right it is Catalase test. So for Catalase test, we need to take few ml of coagulase reagent from rabbit plasma on a clean test tube. So if you add some colonies of Staphylococcus aureus, you can start to see a grain or sand or small clumps in this test tube with coagulase reagent, some small clumps. So that indicates that the organisms are coagulase positive. So Staphylococcus is one of the important organisms which is coagulase positive. So when you compare with other organisms which are coagulase negative, you can see clumps. So other organisms which are coagulase negative, if you inoculate on coagulase containing test tube, you can't see any grain or clumps or sand like granules inside this test tube. So that indicates coagulase negative. Virulent factors. For addition, the Staphylococcus aureus always has clumping factors. We can call them as surface proteins. These clumping factors, they always try to attach to laminins of the host cells, fibronectin, fibrin, fibrinogen and collagen. Fibrinogen, by attaching to fibrinogen and collagen, they can majority of the time causes osteomyelitis. In osteomyelitis, for attaching to fibrinogen and collagen plays a major role. Invasion. Staphylococcus aureus organism uses lot of proteins and exotoxins to enter into the human tissues. One important one is alpha toxin. Alpha toxin can also be called as alpha hemolysin. It can damage the membrane, and most sensitive cells are platelets and monocytes, and the mode of action of alpha toxin is by osmotic lysis. Next one is beta toxin. It is a sphingomyelinase, and on blood agar culture medium it is the one which causes beta hemolysis. It is also a pore forming toxin which can damage the membranes, especially the red blood cells and leukocytes. Delta toxin. It acts like a detergent like action. Staphylococcus aureus and epidermidis, both these two organisms secretes this delta toxin, and we don't know much that this delta toxin is a virulent factor. In outside of the human host, we can see the delta toxin action as a virulent factor, but not in the human tissues. It is not yet proved. Leucocidans. Leucocidans, they act like alpha toxins, and they are also leads to cause beta hemolysis, and they can also cause lysis of leukocytes, and they majorly acts on PMNs and other leukocytes. This leucocidin is a major virulent factor in causing skin infections, especially dermatonecrotic lesions. Dermatonecrotic stains are more potent in causing skin infections and abscesses. Coagulase. These organisms have a coagulase substance on their surface, and it is always, when compared to clumping factors, it is different from clumping factors. Even though it is present on the surface with clumping factors, it is different. Genetically, it is proven. Coagulase enzyme is specifically seen on Staphylococcus aureus. This coagulase enzyme always binds with prothrombin. When it binds with prothrombin, it makes staphylothrombin on their surface. So, by having this staphylothrombin, this organism inhibits phagocytosis from the human immune system. These organisms can also secrete staphylokinase. This staphylokinase can break fibrin. They can secrete protease, lipase, and DNases. These three protease, lipase, DNases can help this organism to penetrate as well as to provide nourishment to these organisms. The last invasive substance it has is fatty acid modifying enzyme, that is FAME. This fatty acid modifying enzyme is always helps this organism in causing abscesses. This organism can secrete exotoxins. One is enterotoxin. This enterotoxin always leads to cause vomiting, emesis. Especially this is the one which leads to cause food poisoning within 2 to 6 hours. So the organisms which are present on the skin, whenever in making the food, if the organism contaminates this food, these organisms can grow on the food material which is left over for a prolonged period before eating. So this organism start to produce enterotoxins in the food material. Whenever that contaminated food with enterotoxin was taken by the humans, within 2 to 6 hours we can see food poisoning, majorly vomiting. Toxic shock syndrome toxin 1. This toxic shock syndrome toxin 1 secreted by the Staphylococcus aureus can act as superantigen. It can hyperstimulate the human immune system, especially the T helper cells, and which releases interleukin 1. Ultimately, because of this excessive secretion of interleukin 1, can hyperstimulate the macrophages and other T cells, which ultimately increases interleukin 1 and tumor necrosis factors in the human body, which ultimately leads to toxic shock syndrome. It is more severe condition when compared to toxic shock syndrome of group A Streptococcus. Exfoliatin toxin. Exfoliatin toxin, which is secreted by Staphylococcus aureus, can damage the cadherins in desmosomal connections. Whenever they damage the desmosomal connection, we can see scale skin syndrome, peeling of epidermal part, especially that from stratum spinosum you can start to see the peeling of skin, discoloration of the skin, which ultimately leaves a red glistening area on the skin tissues. And this scale skin syndrome is majorly seen in children, and it is also referred as diaper rash. They can secrete penicillinase enzyme, which can break the beta-lactam rings of beta-lactam drugs like penicillin. So these organisms are resistant to penicillins, and even they are resistant to other antibiotics even. Host defense escape. This organism escaped from the host defense by capsular polysaccharide. This organism has a very thin polysaccharide capsule, so it can inhibit phagocytosis. This organism can secrete protein A material. This protein A binds to IgG molecules on the Fc region of the IgG molecules, so it inhibits opsonization.
Antigens are normally processed by antigen-presenting cells and presented to T helper cells on class II MHCs. Only the T helper cells with T cell receptors capable of interacting with the antigen are stimulated to produce cytokines. The activated T helper cell stimulates only B cells that react with the same antigen, thereby limiting the response to the production of antibodies that were presented. Superantigens, such as the toxic shock syndrome toxin produced by Staphylococcus aureus, are not processed by antigen-presenting cells. Instead, they bind directly to the outer portion of an MHC class II antigen on antigen-presenting cells, and to the outer portion of the T cell receptor of T helper cells. Rather than binding to only the cells specific for that antigen, which represent about 1 in 10,000 T cells, The superantigen binds without antigen specificity to as many as 2% to 20% of the T cells. This results in stimulation of many T helper cells. The result is the release and activity of excessive amounts of interleukin-2, IL-2, and other cytokines that enter the bloodstream instead of only acting locally as they normally do. This leads to symptoms of fever, nausea, vomiting, diarrhea, and sometimes shock, with failure of many organ systems, circulation failure, and often death.
These are the virulent factors that Staphylococcus aureus organism have. It has protein A microcapsule which is made with polysaccharide. It has a cell wall with lot of invasions, hyaluronase, staphylolysin, leucocidins, leucotoxins, coagulase, staphylokinase. It has lot of clumping factors which can bind to fibrin, fibronectin, ultimately damage the cells. It can secrete toxins, exotoxins, toxic shock syndrome, toxin 1, exfoliating toxin, and enterotoxins. Epidemiology. It is the most opportunistic pathogen. Whenever a human immune system suppressed or human suffers with some kind of systemic infection, this organism takes a lead and causes the infection in humans. It is nosocomial. This organism can be seen in hospitalized environments. People who are suffering with other systemic conditions and who are hospitalized, those individuals are highly get these infections from the hospitals. We can call them as nosocomial infections. We can see this organism on humans in skin, mucous membrane, and nasal areas. Food, we can get the transmission through food, through tampons. Tampons are one of the important factors in causing toxic shock syndrome. Any type of tampons like vaginal tampons, surgical tampons, nasal tampons can lead to cause this infection. Trauma can also lead to cause this infection, and immune suppressions always help this organism in causing infection. This is the diagrammatic representation of the infections that are caused by the Staphylococcus aureus. It can cause skin infections like styes, boils, carbuncles. We can see sinusitis, furuncles. We can see pneumonia by this infection, especially hospitalized patients. Cystic fibrosis patients are most prone to get the pneumonia. And we can have whenever this organism enters into the blood, hematogenous spread can lead to cause endocarditis, osteomyelitis, or septic arthritis. It can cause skin infections like impetigo, emesis, vomiting, food poisoning, majorly because of exotoxins secreted by Staphylococcus aureus. It can cause diarrhea in toxic shock syndrome and urinary infections, scale skin syndrome. And it is one of the major important cause of mastitis, the breast abscess. Pathogenesis, diagrammatic representation of pathogenesis. These organisms can cause skin infections. The major virulent factors are leucocidins and hemolysins. Pneumonia, the major virulent factors are hemolysins, leucocidins, and coagulase. Heart infections, cardiac infections, especially it can cause acute endocarditis, infective endocarditis. The major virulent factors are coagulase, staphylokinase, hemolysins, and leucocidins. It can cause osteomyelitis and septic arthritis. It is one of the most common cause of osteomyelitis and septic arthritis, majorly by hematogenous spread. The virulent factors are surface proteins to collagen, hemolysins, and leucocidins. It can cause food poisoning, especially by enterotoxin, and it can cause toxic shock syndrome by toxic shock syndrome. Toxin 1 and enterotoxin are the virulent factors for toxic shock syndrome. It can cause scale skin syndrome, especially in children, with exfoliating toxin. Clinical features, clinical conditions that are caused by Staphylococcus aureus. It can cause different types of skin infections like folliculitis, carbuncle, furuncle, impetigo, and it is the major cause of one of the most important cause of mastitis, breast abscess, and it can also cause surgical infections when Suture conditions can lead to cause in contaminated sutures can lead to cause the skin infections, and using of catheters, using of urinary catheters can also lead to cause infections in humans. Much opportunistic and people who are immune suppressed are more prone to get these infections. It can cause pneumonia, especially in hospitalized patients or people with post-influenzal viral influenzaal infection or cystic fibrosis. It is not written here, but cystic fibrosis and immune compromised patients. In pneumonia condition, we can see patients suffering with fever, chills, cough, consolidated lungs and pleural effusions and cavity formation of the lungs. It can cause acute infective endocarditis, especially in IV drug abuse patients, and people who have this infective endocarditis by IV drug abuse, majority of the time they have a tricuspid valve involvement, and more vegetative forms are seen on tricuspid valve, and because of desquamation of this group of organisms as an emboli can ultimately leads to cause pneumonia in these patients. In acute endocarditis, the symptoms starts with sudden onset of fever, anemia, you can see roach spots, you can see splinter hemorrhages, you can see Genevieve nodes, you can see oscillus nodes, and the patients have murmurs, and there is no need of having previous cardiac conditions like rheumatic fever or valvular disease or any kind of cardiac condition. Suddenly we can see endocarditis by Staphylococcus aureus without having any previous cardiac problems. Osteomyelitis and septic arthritis is the most common cause of osteomyelitis and septic arthritis, and most common in conditions like people who are having trauma or accident conditions can lead to cause these two conditions, and majority of the time we can get by hematogenous spread. Whenever this organism enters into the bloodstream through skin infections or any kind of sources, whenever they enters into the bloodstream, they can lead to cause osteomyelitis and septic arthritis. We can see the septic arthritis as irreversible type of arthritis when compared with rheumatic arthritis. Food poisoning. We can see food poisoning in this condition majorly because of the enterotoxin, which is already present in the food, contaminated food. So that can lead to cause food poisoning. Especially you can see this condition starting with between 2 hours and 6 hours after having the food, and the majority of the time the patients will have vomiting, and it is self-resolving within 24 to 48 hours. We can see the resolution of this condition. Major sources are mayonnaise and salads. People who are eating this contaminated mayonnaise and salads are people who ate the food which was made a long time back or 6 hours or 10 hours back, can always lead to cause food poisoning by Staphylococcus aureus. It starts with sudden onset within 2 hours with vomiting. Toxic shock syndrome. The predisposing factors for toxic shock syndrome is tampons, sutures, endometritis and unsterile abortions can lead to cause toxic shock syndrome. In toxic shock syndrome, we can see sudden onset of fever, malaise, myalgia, patients having vomiting, nausea, diarrhea, and with rashes all over the body including palms and soles, and ultimately leads to cause desquamation of skin even, and it is one of the fatal condition which leads to shock. Scale skin syndrome. The scale skin syndrome is caused mainly by the virulent factor exfoliating toxin secreted by this organism, majorly seen in children with skin infections or children with umbilical cord infections. And in this condition we can see sudden onset of fever in the patients, and they will have a desquamation of skin, especially the stratum spinosum layer. Up to the stratum spinosum layer will get desquamated, leaving the basal layer, stratum basale. So the patients with scale skin syndrome appears like a reddish glistening areas without bleeding, and it is also referred as diaper rash. So here we can see some pictures of these conditions. This is impetigo caused by Staphylococcal aureus organism with blisters. Whenever the blisters are burst out, you can see the ulcer formation, especially seen in children. and this is scale skin syndrome. You can see the redness, glistening redness, and discoloration of the epidermis up to stratum spinosum layer. And this exfoliating toxin can break the desmosomal connection between the cells so that the upper part of the epidermis will get desquamated. So it gives a redness. Folliculitis, hair follicle infection. Whenever there is a break in the skin, these organisms are always present on the skin. Whenever there is a break in the skin, they can lead to cause the skin conditions. One major one is folliculitis. They can cause furuncles whenever these organism enters deep into the tissues. They can cause abscesses which can drain through sinus. And this is carbuncle. Whenever this organism enters into the subcutaneous tissues, it can cause multiloculated abscesses which can drain through sinus tracts. Carbuncle. And this is toxic shock syndrome. You can see the rashes, reddened rashes on the palms here, and the peeling of skin, and it can lead to shock condition. Sorry. Diagnosis. We can diagnose this organism depending upon gram staining. On a gram staining, we can see this organism in clusters. Gram positive blue color organisms in clusters. They are catalase positive organism. In catalase test, we can see the air bubbles coming from the hydrogen peroxide when we inoculate Staphylococcus aureus organism on hydrogen peroxide. They are coagulase positive. They have this coagulase enzyme on their surface, so they can make clumps or grain or sand-like structures in coagulase reagent in coagulase tests. They are coagulase positive. On blood agar culture medium, they grow as beta hemolytic colonies with yellow color discoloration or golden yellow color pigmentation around the colonies when compared to other staphylococcal organisms. Other staphylococcal organism, you can just see the growth of the organism without having any pigmentation. They are PYR positive, pyrrole idle aryl amidase positive, and they are bacitracin resistant. On bacitracin containing media, these organisms are resistant and they grow on bacitracin containing medium. And they are mannitol positive. When we compare with other epidermidis and Staphylococcus saprophyticus, these organisms can ferment mannitol. That is one of the important diagnostic feature. It is missing here. Gram positive cocci identification. So gram positive cocci always round in nature. Gram positive cocci are always round blue color in nature, whereas bacilli, they are rod shaped. These are the bacilli in gram positive: Corynebacterium, Clostridium, Listeria, Bacillus. Whereas coccis, we have two varieties of coccis. One is Staphylococcus and other one is Streptococcus. Staphylococcus are catalase positive, Streptococcus are catalase negative. If they are catalase positive, in Staphylococcus we have two groups of organisms. One variety is coagulase positive organisms, that is Staphylococcus aureus, one organism, and coagulase negative organisms are Staphylococcus epidermidis and Staphylococcus saprophyticus. So to differentiate this coagulase negative organism, these two, Staphylococcus epidermidis and saprophyticus, we can use novobicin test. On novobicin containing medium, this Staphylococcus epidermidis are sensitive. They won't grow in novobicin containing medium. Whereas Staphylococcus saprophyticus on novobicin containing medium, they are resistant. They will grow in novobicin containing medium. So we can identify the Staphylococcus depending upon coagulase test and novobicin test to differentiate between non-coagulase organisms. Coming to the Streptococcus, they are catalase negative when compared to Staphylococcus. They are catalase negative. They are round the cocci organisms. They are catalase negative. They can be classified in different ways depending upon hemolytic test. In beta hemolysis, beta hemolytic organisms are Streptococcus pyogenes. They are also called as group A and they are bacitracin sensitive. And another beta hemolytic organism in Streptococcus is Agalactiae, Streptococcus agalactiae, that is group B, and it is bacitracin resistant. And gamma hemolytic organisms are Enterococcus. Enterococcus fecalis and faecium, and one more organism is Streptococcus bovis. And alpha hemolytic organisms, they causes partial hemolysis on blood agar culture medium. They look green in color, part of alpha hemolytic organism. One alpha hemolytic organism in Streptococcus is pneumoniae, and it is oprogen sensitive. On oprogen containing media they won't grow, and they are bile soluble. In bile containing media they will get easily distracted, and they have a capsule. So we can also use Pullung's test, and it is Pullung's positive. And another alpha hemolytic Streptococcus organism is viridans Streptococcus. In viridans we have mutans sanguineus, mimetis, salivarius, and it is oprogen resistant. In oprogen containing media these organisms will grow, and they are not bile soluble. They won't get distracted by bile, and they won't have any capsule. So in this way we can differentiate gram positive organisms. Treatment. Staphylococcus aureus is resistant to penicillin because it always have a penicillinase enzyme. And vancomycin is the drug of choice. For majority of the Streptococcus strains, vancomycin is the drug of choice, but few serotypes of Staphylococcus aureus are sensitive to penicillin, but majority are resistant to penicillin and other antibiotics even. MRSA, methicillin resistant Staphylococcus aureus. Some strains of Staphylococcus aureus are resistant to methicillin antibiotics, and there are two groups of MRSAs. One is hospitally acquired. HA refers to hospitally acquired methicillin resistant Staph aureus, and another one is CA MRSA. CA means community acquired methicillin resistant Staph aureus. So these two groups of Staphylococcus aureus organisms are resistant to antibiotics. They are resistant to penicillins. They are resistant to methicillin, nificillin. So they are resistant to major important antibiotics. So people who are susceptible for these infections are hospitalized patients. These organisms, majority of the time, this HA-MRSA is seen in hospital environments, but nowadays we are seeing in community also. That's why there is community acquired strains of this organism. Hospitalized patients are susceptible for these MRSAs. Immune suppressed individuals and improper hygiene patients with especially like athletes who are going for workouts and after coming from workouts who won't take shower, they are most susceptible. With bad hygiene they are most susceptible with these community acquired MRSAs. These organisms are always present on human tissues, on skin, on areas and mucous membrane areas. And the treatment for these organisms is vancomycin. Vancomycin is the drug of choice, but nowadays we are observing vancomycin resistant MRSA organisms, methicillin resistant Staph aureus organism. For those organisms nowadays we are using one more drug called quinipristone. Staphylococcus epidermidis. These organisms are gram positive in clusters, and they can't be differentiated with Staphylococcus aureus in gram staining. They look similar in gram staining. They are coagulase negative. They are mannitol negative. They won't ferment mannitol when compared with Staphylococcus aureus. Coagulase negative and mannitol negativeness is the primary differentiating feature between Staphylococcus epidermidis and Staph aureus. They consist of thick polysaccharide capsule. This polysaccharide capsule is the main virulent factor for this organism. It helps in adhesion, it helps in restricting the phagocytosis, and it helps in attaching to the prostheses that are present inside the human body, like the joints, artificial valves and pacemakers, etc. And they can lead to cause prosthetic infections. These organisms can also secrete delta toxin, which acts like a detergent function. These organisms are most commonly seen on skin. When compared to Staphylococcus aureus, these are most commonly seen on skin than Staphylococcus aureus. And these are the most common contaminants of blood samples because they are widely seen on skin. They are most common contaminants. So whenever we are diagnosing these organisms, we need to take 2 or 3 samples to confirm the diagnosis. Blood samples from different regions of the human body to confirm diagnosis. These organisms can cause nosocomial infections. They are resistant to antibiotics. Virulent factors of Staphylococcus epidermidis. They have a polysaccharide capsule, thick polysaccharide capsule which is gelatinous in nature. So with the help of this polysaccharide capsule, these organisms which are present on the skin can enter into the bloodstream with catheters or IV lines or urinary catheters. They can enter into the bloodstream and they can go and attach with the help of polysaccharide capsule with the prosthesis or prosthetic joint areas or the prosthesis inside the heart, like artificial valves or pacemakers. They will go and attach there and they can cause infection. And it has delta toxin which have a detergent activity, so it can break, it can make pores on the cells of human tissues, so it can lead to the destruction of cells. Clinical conditions. It can cause nosocomial infection because IV lines, catheters and all these urinary catheters, endoscopic tubes, all these instruments are used in hospitalized environment. There is a high chances of entry of this organism into the bloodstream in hospitalized environments. So we can have nosocomial infections. Catheters and prosthetic devices are major source of these infections. So catheter infection and prosthetic devices infections are majorly seen because of Staphylococcus epidermidis. And staph aureus can also lead to cause the surgical infections like catheters, but this is one of the major important one in causing these infections. It can cause endocarditis of prosthetic heart valves. Whenever this organism enters into the bloodstream and attaches to this prosthetic heart valves, it can lead to cause endocarditis, and we can see the symptoms of endocarditis which we discussed previously. It can cause artificial joint infections. Any processes that is present in the human body can be infected by this organism. Diagnosis. These organisms are coagulase negative, mannitol negative. They are beta or gamma hemolytic on blood agar. You can't see any yellow color pigmentation or yellow color discoloration on blood agar culture medium. Novobisin sensitive when compared to epidermidis. These organisms won't grow in novobisin containing medium. Treatment. These organisms are resistant to antibiotics because they are always seen in hospital environments. They are resistant to antibiotics, and the vancomycin is the drug of choice for Staphylococcus epidermidis. Staphylococcus saprophyticus. These organisms are gram-positive coccine clusters. They are non-hemolytic. They are always gamma hemolytic. They are coagulase negative, mannitol negative, and they are novobisin resistant. When compared to Staphylococcus epidermidis, these organisms can grow in novobisin containing medium. These organisms causes urinary tract infections in suddenly sexually active women, and this can be referred as honeymoon cystitis. And this infection can be normally seen in women who are newly married. We can see this urinary tract infection, especially urinary bladder infection. In this condition, the patient will suffer with dysuria, pain during urination, and they'll have increased frequency of urination. Diagnosis. Novobisin resistant Staphylococcus saprophyticus is novobisin resistant. It can grow in novobisin containing medium when compared to Staphylococcus epidermidis. It is coagulase negative, mannitol negative, and the treatment, penicillin is the drug of choice. We can use penicillin drug for this organism. It is not resistant to penicillin. We can use penicillin for this organism.